Hi everyone, and welcome to Lab 3, Extracting DNA. In the previous lab, you learned about the structure of the typical eukaryotic cell, the cells that comprise the human body. You learned that most body cells have three main parts: a nucleus, cytoplasm and organelles contained within it, and the plasma membrane. During the discussion of the cell, we focused quite a bit of attention on the nucleus, and in addition to being the control center of the cell, we said that the nucleus is important because it houses DNA, the molecule of heredity. In this lab, you'll learn more about DNA, examining its microscopic structure. Afterwards, you'll complete a hands-on experiment in which you attempt to extract DNA from cells to see if it's possible to look at DNA without the use of a microscope. Before looking in more detail at the structure of DNA, let's quickly recap what you learned last week about the nucleus. Remember that the nucleus is the largest organelle in the cell, and the contents of the nucleus are separated from the rest of the cell by a double-layered membrane called the nuclear envelope. Remember we said that the double-layered membrane is important because it separates DNA from the rest of the cell, ensuring that it doesn't get damaged. Now remember, in this particular figure, DNA is in the form of chromatin, highlighted with the red arrow. Remember we said that chromatin is a very thin, thread-like form of DNA. And as you look at the figure, you can see lots of dark purple strands, each of which represents a molecule of DNA. But when you think of DNA, this string-like structure is probably not the image that comes to mind. And when you think of DNA, typically the structure you picture looks more like this. You should remember from general biology that DNA resembles a ladder that's been twisted, giving it a helical shape. This helical shape is one defining characteristic of the DNA molecule. As we move on, we'll look more closely at the helical structure of DNA, focusing on the building blocks of DNA that connect to form the DNA double helix. At this point, you should know that DNA, like all biological molecules, can be categorized into one of four main groups. The four main classes of biological molecules are carbohydrates, lipids, proteins, and nucleic acids. So the question is, which of those four classes does DNA fit into? If you said carbohydrate or protein, you're wrong. DNA rather is a nucleic acid, and like all nucleic acids, DNA is composed of nucleotides. All nucleotides have three main components. All nucleotides have a 5-carbon sugar, a nitrogenous base, and a phosphate group. The figure you see here shows the basic structure of the nucleotide. Take a moment to identify the three major parts of the nucleotide. In the center, you see a sugar molecule color-coded with blue, a phosphate group in yellow, and a nitrogenous base in green. Both the sugar and nitrogenous base that make up the nucleotide are organic molecules. Remember that a molecule is classified as organic if it contains the element carbon. Now, I know most of you have not taken organic chemistry, so when you look at organic compounds like this sugar and the nitrogenous base, it seems a little foreign to you. And although you've not taken organic chemistry, it's still important that you're able to recognize organic compounds and understand their structure. Okay, let's use the sugar molecule as an example. Okay, first, let's separate the sugar molecule from the phosphate group and nitrogenous base. Now we can get a better idea of what this molecule looks like. Now, this particular sugar found in DNA nucleotide is called deoxyribose, hence the name deoxyribonucleic acid, for which DNA is short. Most of the organic compounds you'll see in your textbook are drawn with this sort of geometric shape, and this particular molecule has a pentagonal shape. Now, remember we said that the sugar that comprises the DNA nucleotide is deoxyribose, and it is a 5-carbon sugar. Okay, although you only see one carbon atom labeled in this figure, there are in fact 5 total carbons. Everywhere you see two sides of this pentagonal shape meet, there's a carbon atom. For example, where you see the red arrow, two sides of the molecule are connected, and at that point, there's a carbon atom. You can locate the remaining carbons by starting at this point and working your way clockwise around the pentagonal sugar. Now, all of the carbon atoms that comprise this sugar must be labeled. We have to keep track of which carbon atom is where. For example, the first carbon you see indicated by the red arrow is called the 1' carbon. Moving around the molecule, you see the second carbon, which is called the 2' carbon. The third carbon is called the 3' carbon. There is the fourth carbon called the 4' carbon. And finally, the fifth carbon is called the 5' carbon. Hopefully you remember from lecture that carbon atoms form a maximum of 4 bonds with other atoms. And if you take a look at each of the 5 carbon atoms in the deoxyribose sugar, you should notice that each of those carbon atoms has 4 total bonds. For example, the 3' carbon is bonded to the 4' carbon, the 2' carbon, to a hydrogen atom, and to an OH group. The OH group that is bonded to the 3' carbon is called a hydroxyl group. Before moving on, go around the sugar molecule and convince yourself that each of the carbon atoms has formed 4 total bonds. Alright, now let's get back to the topic of labeling each of the carbon atoms in the sugar molecule. You may be asking yourself, why does it matter? Why does it matter which carbon atom is which? Why, for example, didn't we start on the left-hand side and work our way around the molecule counterclockwise? The way in which these carbon atoms is labeled is important, especially when we start looking at how individual nucleotides are linked together to form an entire DNA molecule. If you would, focus your attention back on the nucleotide figure on the left side of the screen. Notice that the 1' carbon is attached to the nitrogenous base, and the 5' carbon is attached to the phosphate group. This method of attaching the nitrogenous base and the phosphate group to the sugar is the same for all nucleotides. So, in other words, the nitrogenous base is always attached to the 1' carbon, and the phosphate group is always attached to the 5' carbon. Again, this is something that's very important to remember when we start looking at how nucleotides combine to form the DNA molecule. In the next figure, you can begin to see what happens when nucleotides link together to form the helical DNA molecule. Again, remember from the figure on the previous slide, the sugar molecule was outlined in blue and the phosphate groups were highlighted in yellow. And you see here that the backbone of DNA molecule, in other words, the side of the DNA ladder, is composed of the sugar and phosphate groups. Notice that the nitrogenous bases of each nucleotide extend out to the center of the DNA molecule. It's the nitrogenous bases that form the steps or rungs of the DNA ladder. Also notice in this figure that there are 4 different nitrogenous bases, each represented by a different capital letter. A represents the base adenine, T, thymine, C, cytosine, and G, guanine. Let's take a closer look at each of the 4 nitrogenous bases found in a DNA molecule. The left-hand side of this figure shows the structures of the 4 nitrogenous bases found in DNA. The upper 2 bases highlighted with the red arrow are the pyrimidines, and they include thymine and cytosine. The 2 bases at the bottom, adenine and guanine, are purines. Take a moment to compare the organic structures of the purines and pyrimidines. Notice that the purines have a slightly different organic structure than the pyrimidines. The purines are bigger molecules than the pyrimidines. For example, the pyrimidines are a single-ringed structure with 6 sides. whereas the purines consist of a 6-sided carbon ring fused to a 5-sided carbon ring. Now, for lab, it's not necessary that you memorize the organic structure of each of the 4 nitrogenous bases. So you'll never see a question in which you're given a picture of adenine and asked, is this adenine, guanine, thymine, or cytosine? You should, however, be able to identify a nitrogenous base as being either a purine or a pyrimidine. Now, the way I like to keep each group separate in my mind is to think about the length of the words purine and pyrimidine compared to the size of the molecule. So, for example, the pyrimidines, thymine and cytosine, are single-ringed structures. So they're smaller than the purines, but the word pyrimidine is much bigger. In other words, it has more letters in it. So here we have a big word and a small molecule, whereas with the purines we have a shorter word and a larger molecule. So again, you should be able to identify a nitrogenous base as being either a purine or a pyrimidine. You should also remember which bases fall into which category. Again, thymine and cytosine are the pyrimidines, and adenine and guanine are the purines. The RNA nucleotide in the right-hand side of the figure is something that we'll come back to and explore in more detail later on in the semester. But what you should notice is that it has the same basic structure as the DNA nucleotide we saw earlier. It has the same parts, including a 5-carbon sugar, a nitrogenous base, and a phosphate group. The figure you see here shows you the basic overall structure of a DNA molecule. Again, remember that DNA has a twisted ladder shape, and the outer sides of the ladder are composed of the sugar and phosphate groups of each nucleotide linked together. The steps of the DNA ladder are formed by the nitrogenous bases that protrude into the interior of the DNA molecule. Notice in this figure the dashed line that connects each of the nitrogenous bases. The dashed line represents an attractive force between those bases, and it's that attractive force that holds the sides of the DNA molecule together. In the center of the figure, notice the highlighted AT base pair. As you look along the entire DNA molecule, you can see that adenine and thymine are always paired together. Likewise, cytosine and guanine are also always paired together. Okay, this is true of all DNA molecules. Now, mutations in DNA may result in a mismatched base pair, but at this point we're not going to concern ourselves with DNA mutations. So for now, keep in mind this basic structure of DNA and the general rule that adenine and thymine always pair together and cytosine and guanine always pair together, and those bases are held together by attractive forces. But the question to ask at this point is, what kind of attractive forces hold those bases together? Okay, take a look at the next figure. Okay, here you see a slightly different version of the DNA molecule. Notice that you can still see the dashed lines connecting the nitrogenous bases in the center of the molecule, and those dashed lines represent hydrogen bonds. You should remember from lecture and from general chemistry that individual hydrogen bonds are very weak. But the more hydrogen bonds added together, the stronger the overall attractive force. Okay, remember that hydrogen bonds are what give water its extraordinary properties. Okay, notice in the figure that adenine and thymine are held together by 2 hydrogen bonds, and cytosine and guanine are held together by 3 hydrogen bonds. So which base pair is stronger, an adenine-thymine base pair or a cytosine-guanine base pair? Hopefully you recognize that the cytosine-guanine base pair is the stronger of the 2 base pairs because it has one additional hydrogen bond that the adenine-thymine base pair does not have. So the more hydrogen bonds there are, the stronger the overall connection. Okay, at this point we've talked about the base pairing that occurs between the nitrogenous bases in the center of the DNA molecule. But I want to go back and look once again at the sugar-phosphate backbone, which forms the sides of the DNA molecule. Before we do that, however, let's return to the structure of the nucleotide. Again, remember it's the sugar and phosphate groups of the nucleotide that form the sides of the DNA ladder. As you look at the nucleotide in the figure, focus on the phosphate group. Okay, there's something very important about the phosphate group that should jump out to you. In looking at the phosphate group, hopefully you notice that one of the oxygen atoms in the group has a negative charge on it. Because that oxygen atom has a negative charge, the phosphate group as a whole is negatively charged. If the phosphate group, which is one of the components of the DNA backbone, is negatively charged, then what does this mean about the charge on the DNA backbone itself? Well, because one of the components of the DNA backbone is negatively charged, the DNA backbone overall is also negatively charged. So a DNA molecule as a whole is a negatively charged molecule because the phosphate groups which comprise part of its backbone are negatively charged. Now, this is a concept that's very important for you to remember when it comes time to understanding what happens in the hands-on experiment you'll complete later on. You have to remember that DNA is negatively charged to understand the process involved in extracting it from cells. Alright, now let's take a closer look at the structure of the DNA backbone. If you would, focus your attention on the sugar-phosphate backbone in the center of the figure. Notice that the backbone is highlighted by a light blue rectangle. Okay, in this figure, several nucleotides have been linked together, forming a DNA backbone. Okay, to simplify the figure, focus on the nucleotide in the center, surrounded by the white block. Again, notice with this nucleotide, you can see the three main parts, including the phosphate group represented by the yellow circle, the sugar in blue, and the nitrogenous base, which in this case is thymine. Okay, recall that before we identified and labeled each of the carbon atoms that comprise the deoxyribose sugar. So here is the 1' carbon. Remember that the nitrogenous base is always attached to the 1' carbon. This carbon atom is the 3' carbon. And you can also see the 5' carbon to which the phosphate group is attached. Now remember before I said it's important for us to be able to recognize and identify each of the 5 carbon atoms that comprise the sugar when it comes to understanding how nucleotides are linked together to form the DNA molecule. So again, if you would, focus on the phosphate group attached to the nucleotide in the white box. And now look at the nucleotide that's directly above it. Okay, which carbon atom is the phosphate group on the nucleotide in the box connected to on the nucleotide above it? You should notice that the phosphate group is connected to the 3' carbon of the nucleotide above it. And as you look at the remainder of the DNA backbone, you should see the same pattern exists all the way up the backbone. Okay, the 5' phosphate of one nucleotide is linked to the 3' group on the sugar above it. Okay, this pattern of nucleotide binding is the same for all nucleotide molecules, and we refer to this pattern as the 5' phosphate to 3' sugar link. Take a moment and make sure you can identify each of the 5' phosphate to 3' sugar links in the figure. Now, if you would, focus your attention on the two ends of the DNA backbone. You should notice that the two ends look different. At the top of the strand, you see a phosphate group, and that phosphate group isn't attached to another nucleotide. At the bottom of the backbone, you see a sugar group with its hydroxyl group, which isn't attached to another nucleotide. Okay, to further illustrate this, take a look at the next figure. Okay, first look at the left-hand side of the figure. Again, you should notice that this half of the DNA molecule has the same structure as the one we saw on the previous slide. At the top of the DNA backbone on the left, there is a free phosphate group, which remember is attached to the 5' carbon. At the opposite end of the molecule, there is a free 3' hydroxyl group. Because the two ends are different, we can give them a specific label. Okay, the end with the free phosphate group is called the 5' end, and the end with the free hydroxyl group is called the 3' end. Now compare the left side of the DNA backbone with the right-hand side. You should notice that the DNA backbone on the right looks like a mirror image of the left-hand side that's been flipped upside down. So in other words, on the right-hand side, the free phosphate group is at the bottom of the strand and the free hydroxyl group is at the top. So in this particular strand, the 5' end is at the bottom and the 3' end is at the top. Because the two sides of the DNA backbone are different, in other words, because the strands run in opposite directions, the two strands are referred to as being anti-parallel. Now you're probably wondering, why does it matter? Okay, why do we care that one strand runs from the 5' end to the 3' end and the other one runs from the 3' end to the 5' end? We care because when it comes to understanding the processes of DNA replication and protein synthesis, we have to understand the structure of the DNA backbone before we can understand how those processes take place. So for now, remember that the two backbones that comprise the DNA double helix have opposite orientations. One runs 5' to 3' and the other runs 3' to 5'. One final thing I want to mention is that although the two DNA strands have opposite orientations, we still see the same basic pattern of nucleotide connection. In other words, the 5' phosphate is linked to the 3' sugar of the adjacent nucleotide. That concludes the Lab 3 podcast on DNA. At this point, you should follow the instructions outlined in your lab handout for completing the hands-on experiment, extracting DNA.